Hey, thank you. So I'd like to thank the organizers for inviting me to give this talk. And I'm also honored to be speaking next to or along with the other esteemed speakers here. So I'm going to tell you about the work my lab has done in which we're imaging the cellular plasma membrane. So the plasma membrane is a selectively permeable barrier that surrounds cells. It's composed of a lipid bilayer, so that's shown here in this cartoon. So there's many different lipids. I have shown here the lipids as blue and purple as, well, that only shows two, but there's hundreds of different lipid species. There's cholesterol, which is a sterol. There's transmembrane proteins that are responsible for a lot of the functions. And some of these proteins, as well as lipids, are glycosylated. So those are long sugar molecules on the surface of the cell. So the initial lipid membrane model was thought to be homogeneous. So this lipid membrane the lipids are free to diffuse in the plane of the bilayer. They flip rather slowly. And this motion was thought to produce a cell membrane that is pretty homogeneous in terms of its lipid composition. However, as time went on, observations were made that led to the hypothesis that cholesterol, which is shown here, and a type of lipid called a sphingolipid. So here in this kind of orangish color, these are all sphingolipids. They have the same backbone of the lipid, but they could have all different head groups on it. It was thought that this cholesterol and sphingolipids, well, they have favorable interactions, at least in model systems, that cause them to co-localize together and form domains. It was thought that these domains that are enriched with cholesterol and sphingolipids, which have been called lipid rafts, it was believed that they exist in mammalian cells and that they're important to function, that they recruit certain proteins to them, and that close interaction between the proteins gives rise to function in the cell. These lipid rafts have been difficult to detect, though. There has been, at least if you try to label the cholesterol and sphingolipids and look at them with something like fluorescence microscopy, there were a range of conflicting results, and it was thought that the fluorophores were just as big as the lipids. And since molecular interactions that are non-covalent were responsible for function, that those dyes were messing up the interactions and making it unreliable as a technique to look at. So that's where we came in, and we wanted to apply the nanosims imaging in order to detect the lipids. So what is relevant to these experiments, I know that you've already heard a description of the nanosims. So I'm pointing out here what's important for our experiments to keep in mind. So the lateral resolution is as good as 50 nanometers. There's been reports of it being even better. So that's great because these lipid domains were thought to be under 200 nanometers. This is performed under ultra high vacuum, so we have to chemically fix the cells. That was just mentioned in some questions that Matt answered for us. So I'll have a little more on that on the next slide. What's really important too is that the sampling depth could be limited to the plasma membrane. And that's important because there are other membranes beneath the cell membrane. So there are vesicles and organelles that could be beneath it. And so the issue is if you detect an excess of sphingolipids, is that because they are concentrated in the plasma membrane, or is that because you detected two membranes, so something beneath the cell membrane as well? And then, so we're using a NanoSims 50, not the 50L instrument. So the NanoSims 50 has 5 different mass to charge or detectors, 5 different detectors. So we could detect 5 different mass to charge ratios. And that is monoatomic and diatomic species, so fragments of the molecules of interest. So that means if you're getting monoatomic and diatomic secondary ions, you're getting elemental or isotopic composition, and hence we need to isotopically label our samples. Also important is a high mass resolving power. so we could discriminate between interfering isobars of the same nominal mass. So in plain English, that means we could tell things apart that has approximately the same mass to charge ratio. So carbon-12, nitrogen-15, where nitrogen-15 is a rare stable isotope of nitrogen, has a mass of 27. And so does carbon-13, which is a rare isotope of carbon, and nitrogen-14, where nitrogen-14 would be the abundant isotope. So they have approximately the same nominal mass, but we could have no problem distinguishing them from one another with the nanosims. So to apply this technique to look at cell membranes, and I'm going back to our first experiments on cells, um, in which we started by looking at just the sphingolipids. In order to incorporate rare isotopes into the sphingolipids, we feed the cells, what's shown here is a metabolic precursor of sphingolipids. So this molecule, it's a sphingoid base. So cells will take it up out of the medium and use it to synthesize their own sphingolipids. These are mammalian cells, so they have limited metabolic pathways. We could perform this under conditions that the rare stable isotope does not wind up in other lipids. Basically, we just feed it to the cells. It's kind of like feeding tropical fish. If you've ever had tropical fish or marine fish in a fish tank, if you give them too much food, you kill them. If you don't give them enough, you kill them. So there's a perfect balance that is pretty straightforward to optimize. So we just add that to the culture medium. And in these experiments, we were also feeding the cells uniformly labeled carbon-13 fatty acids. So by uniformly labeled, it's that all the carbons along the chain contain the rare stable isotope of carbon, C13. And I have shown one structure here. There were a variety of structures, which means that they had different degrees of saturation, different lengths. And that was so this carbon-13 labeled fatty acids wound up in all the lipids. That was so we could assess if we found an excess of sphingolipids. Is that, again, because they were concentrated in the plasma membrane, or it didn't mean we were detecting excess lipid? So those two are added to the cell culture medium for about a week under low concentrations. After that time, we chemically fix the cells with glutaraldehyde and osmium tetroxide, so that cross-links all the proteins and lipids so they don't move around when we dehydrate the cell. And then we coat the whole thing with iridium. So that's a metal that makes our sample more conductive and gives us good secondary ion signals. In this process, the cells are intact. When we do our imaging, we do collect secondary electron images that show the cell's morphology, so we know what we're looking at. This technically would be MIMS, according to Matt's description, because we're ratioing the rare stable isotope that's specific to each component, C13 for lipids and 15 for our sphingolipids. The rare isotope is ratioed to the counts of the more abundant isotope that's detected at the same pixel. This allows us to control for the effects of topography, charging, and other factors that are not dependent on lipid composition on our sample. So when we do this imaging, so this is a montage of secondary electron images collected with the nanosims. We see the cell is intact. The extensions are intact. So like I said, this is a whole cell. It's not sectioned in any way. It was grown on a silicon wafer, but we could see it's present. This looks good. So it was well preserved. Here's a carbon-13 enrichment images. So carbon-13 enrichment is encoded at every pixel in color. So dark blue is low enrichment. White would be very high enrichment. So this reddish pink is, about 30 times natural abundance. What's important here is although we do have some pixelation, that's from it's not a super high yielding secondary ion fragment. We do see that this is fairly uniform. We also perform statistical analysis on all the images to confirm it is in fact uniform. You could see some of these micro extensions. So there they were here. They are present, so we could see them. So it is plasma membrane's intact. We don't have any spots that are, large spots missing lipid. If we have this level of pixelation in our sphingolipid images, it's just noise. So that's something to keep in mind. And then, so let's see, what does a nitrogen 15 from our sphingolipid show us? It's right here. So we have a background on the cell of fairly low enrichment. Well, it's enriched compared to natural abundance, but it's not super high. But then within it, there are small domains of high enrichment. Like I said, we do statistical analysis to confirm that this is real and it's not artifacts of our technique. So anything above 12 in this image, which is marked here, so anything that's red, yellow, or orange on the cell, that is a statistically significant elevation in sphingolipids that cannot be attributed to noise. And if we look closely at, zoom into one of these regions on the cell, we see sphingolipid domains and the same location in the C13 enrichment image that was acquired in parallel does not show localized enrichment of C13. So that means all the lipids are not enriched at these sites. We're not detecting excess lipid. We are only detecting um, the plasma membrane here. So we did a lot of control experiments on this. We have found that we've ruled out that it's membrane-bound organelles or vesicles, so it's not excess lipid. It's not any type of aggregation of our labels or lipids due to fixation, which we did with some live cell imaging of a fluorophore labeled sphingolipid. It's not non-specific absorption of our isotope labels on the sample, and it's not the topography. So having rolled out all that, I'll show you briefly, well, what happened with our actual imaging. We said we wanted to look at cholesterol and sphingolipids. Here we have a cell that we fed O18 labeled cholesterol as well as the sphingolipid precursors. So that way the sphingolipids would contain nitrogen 15. We could see them with the nitrogen 15 enrichment images. And our O18 cholesterol, cells have receptors to take that up out of the medium. So that puts the O18 rare isotope into the cholesterol, and we could look at O18 enrichment to see cholesterol distribution on the cells. And so we did the same preservation and iridium coating and same imaging parameters. This is actually a low dose secondary SEM image, secondary electron microscopy image that we took prior to doing the nanosims analysis. It's very low dose. It was like less than 1 kV, which is why it's so lack of contrast. I'm showing you this because we didn't have the secondary electron detector turned on at the time, so I can't show you what it looked like during the nanosims, but we imaged approximately this location. And if we look at that, once again, we have local domain regions of sphingolipid enrichment. So on this cell, it was about 16.3 was statistically significant. So these large regions where it's yellow or orangish, that is regions that denote sphingolipid enriched domains in the plasma membrane. And if we look at O18 enrichment from the cholesterol, we actually see it's fairly uniform. So this region here, this is our cell. We do have overall enrichment of O18 on the cell, meaning we do have O18 labeled cholesterol in the membrane. But the speckling is actually, the speckling here is due to just Poisson statistics of detection. It is not locally elevated. It's not statistically significant enrichments at these single pixel regions that are a little bit brighter. And there's since these images are taken in parallel, we could tell that the sphingolipid enriched domains, the same sites are not, they do not contain statistically significant elevations in cholesterol and sphingolipids. And I know I'm running out of time, so I'll briefly say we've done this on other cells. In this case, this is Madden-Darby canine kidney cells, and I'm mainly showing this. So here's a cell where this is a montage of secondary electron images that were acquired from the cell. This is the secondary electron, or the 15N enrichment image that shows our sphingolipid distribution. I want to point out where the pixels are black, that's where we have no signal at all. The cell was really tall and it was charging, so we didn't, we set a threshold. We didn't exceed the threshold for the minimum count at these sites that allowed us to make significant conclusions about these regions, but it does contain sphingolipid domains. And what I wanted to point out here is sometimes you see, well, often you see things you weren't expecting. And here what we're seeing, this is the overlay of the enrichment image. So again, blue is low enrichment, yellow is high enrichment, showing us where our sphingolipids are. If we look at the edge of the cell here, so I'm zooming in on some of these things. This sphingolipid enrichment, first of all, these like little spots that are on, they're actually on the substrate. And when they're on the substrate, they're pretty round. Often they're at the end of these little extensions. So it actually looks like what we're seeing is sphingolipids that were somehow released by the cell. We believe that would be in the form of some sort of vesicles that are being released at the edge of the cell. So we were not expecting to see that, and we did not see that at all in the our images of the in all the time that we've looked at mouse fibroblast cells. We had not seen that. Cholesterol, it looked the same as what we saw before. It's just uniformly distributed on the surface of the cell. And we didn't see too much in terms of it being secreted as well. I am gonna, well, I'll say we did look at what was causing the sphingolipid domain, since we found it was not cholesterol enrichment. It had to do with the cytoskeleton. If we disrupted the cytoskeleton with drugs, those sphingolipid domains went away, as shown here. And I want to end on real quickly. We have also looked at sphingolipids inside of cells and cholesterol distribution within cells. This is related to disease. I'm not going to go too much into that in interest of time, but to look at inside of the cell, what you do is you could take a series of images at the same location. Each image, since material is sputtered off the cell each time you take a new image, each image is slightly deeper in the cell than the one before. You could keep imaging and sputter away the cell. As you can see here, we started, and then, so we're right about here on the cell. This is a whole SEM image. As we keep taking images, it's getting eroded away. And we can see where this is the cholesterol enrichment as a function of depth, and N15 enrichment would show as sphingolipids as a function of depth. You could reshape these images. You just render them to make a 3D image plane. The problem is if you're doing a sample that has contoured, the image stack is distorted in the Z direction. What I mean by that is since it's a surface sensitive, anything on the surface of the cell comes out at the top of the image, even though in the Z direction, it might be adjacent to the substrate. So here's that picture of the cell. This right down here is the bottom of the cell. This is adjacent to the substrate. This would be the top, but this is coming out at the top because there's nothing above it. So the secondary ions are generated from that region, starting with the first image acquired. I will just say we have developed a depth correction strategy that solely uses what you collected with your NanoSims instrument to reshape those images so you could actually follow the contour of the cell. We recently had a paper on it, and all of the MATLAB code is publicly available on GitHub. So in conclusion, we found that actually that lipid raft hypothesis, it turned out not to be accurate. Cholesterol and sphingolipids were not forming domains in the cells. There were sphingolipid domains that seemed to be controlled by the actin cytoskeleton. So we found that hypothesis to be incorrect actually, and that sphingolipids really were the only thing that was compartmentalized. And we also developed a depth correction strategy that allows you to look at cell distribution within cells and orient it properly so things have the shape of the organelles that a cell biologist or structural biologist would expect, which makes it much easier to collaborate with them and to discuss the results. So with that, I'll thank everybody who contributed to this work and over the years and our funding agencies. And with that, I will stop. And if there's any time left for questions, I'd be happy to answer them.
Yes, thank you very much, Mary. Very interesting. I wanted to, maybe Michiel, do you have any idea about questions being in the Q&A?
We have 3 questions.
Okay, so please go ahead.
I will just ask them. The first one was, how can you prove that 18O corresponds to cholesterol and not 18O from cholesterol that is metabolized into other metabolites?
Because these are not liver cells, they are mammalian cells, so they're unable to break down cholesterol at all. We also did control experiments in which we did traditional analysis, LC mass spec of our cells after they were metabolically labeled to confirm where our labels are going. And they are not winding, at least O18 cholesterol is not winding up in any other cell membranes.
Okay. Thank you. Then another question. Why did you choose iridium to coat your samples? That's the question I also had. Like normally we use gold or...
So when we did this, the bottom line is it was happenstance initially. So the metal coater that we were using was at Lawrence Livermore National Lab. That's where we use the NanoSims instrument, and they happened to have the source in it, what happened to be iridium. Now, iridium, they were using iridium instead of gold palladium because iridium makes a finer grain structure than gold palladium. Both gold palladium, the islands that form when you first deposit it on the sample, and iridium, they're below the diffraction limit of nanosims anyway, or the limit of detection. You're talking, sub 10 nanometer structures. So that's not relevant. That's not important for what we were doing. I think they were using it because they were doing correlated like EM studies, electron microscopy studies where that would be relevant. But we also have done some work where we wanted to detect immunogold. So that's antibodies attached to gold particles. And if you coat your sample with gold, you can't detect your gold particles. So we just stuck with it after that.
Okay. One last question is, why are there latches, steps on the side of the cell?
Why were there what?
These latches or steps.
That's just the way they look once they're fixed, once they're chemically fixed, that particular cell. I mean, it's chemically fixed and dehydrated, right? So that's just what their structures look like when we look at them up close.
Okay.
And Mary, I have one question. So you are talking about imaging of molecules, and this is exactly where people start asking, so how many molecules do we actually see? Do you have a feeling about this, like a ballpark, like if you see an enrichment, for example, in this 15N or this 18O in the cholesterol images you were showing us, do you have a feeling like how many molecules would we be seeing?
Do you mean molecules as in like how many molecules of O18 cholesterol, or do you mean of...
Yes.
Okay. No, I have no idea. I don't know.
Because it's going to this question also, what is the sensitivity of this? And this we are always asked this question. Oh, if you add an isotope and then it's a molecule here, molecule there. Well, can we detect it? And we always don't really know what to say.
It's hard to say because that factor, it depends on the environment that you're detecting it in, right? So if your sample's charging, obviously you're going to get back less signal, and that determines your detection efficiency as well. So I do not know. I know that like we do our metabolic labeling that we're trying to get, say, 50%. at least, of all of the molecules to contain our label.
Mhm.
And I know that we have done experiments where we've done less than that, and we could still detect it. Like, I think as low as 10% we have no problem detecting, but that's more of a pragmatic thing because it doesn't tell us about how many molecules are in the
Yeah.
focal area, right?
Yeah. Okay. Yeah. Okay, but still this is an open question, but okay, thank
Yeah.
you for the answer. So thank you for your talk. Now I think we should be moving on to Silvio.